Dr. Emily Cox is a lecturer in clinical exercise physiology at the University of Newcastle on the Central Coast campus. Emily completed her PhD in 2020 at the University of Queensland, exploring the short and long-term efficacy, safety, and feasibility of a novel low-volume combined aerobic and resistance high-intensity interval training protocol, particularly in people with type 2 diabetes. She's an accredited exercise physiologist, and her research is centered on understanding and influencing the role of exercise in the prevention and management of metabolic disease. In particular, Emily's focus on efficacy and translation and looking at, what types and amounts of exercise are best and how we can get people adopting and adhering to optimal levels of self-directed exercise in the long term. So thank you once again, Emily, for joining us today, and I'll hand the time over to you to talk with us about reducing arterial stiffness in people with type 2 diabetes. Should we target fitness or fatness?
Today I'm going to be talking to you about reducing arterial stiffness in type 2 diabetes and whether we should be targeting fitness or fatness to do that. Before I start, I'd just like to make an acknowledgement of country. So I acknowledge the Darkinjung people, who are the traditional custodians of the land from which I'm presenting today. I would also like to acknowledge the traditional custodians of the lands from which you're all attending. I pay my respect to elders past, present and emerging, and acknowledge any Aboriginal and Torres Strait Islander people attending today. So cardiovascular disease is the leading cause of death in people with type 2 diabetes, and this group experience a 2 to 5-fold greater risk of cardiovascular disease than the general population. Arterial stiffness is a subclinical marker of cardiovascular health and is one of the earliest signs of adverse changes to the vasculature. Because of this, it's an important indicator of cardiovascular risk beyond traditional risk factors such as blood pressure and cholesterol profile. In the absence of disease, arterial stiffness increases 2.5-fold across the lifespan. Previous researchers have suggested that both aerobic fitness and body fatness are determinants of cardiovascular disease risk and mortality because both are associated with greater arterial stiffness. So these variables may represent useful therapeutic targets for the prevention of age-related increases in arterial stiffness in people with type 2 diabetes. But low fitness and high fatness often go hand in hand. So it's unclear whether one of these exerts a stronger influence on arterial stiffness than the other. So that led us to our study, which and the aims of which were twofold. So firstly, we wanted to determine the association between age and arterial stiffness in people with type 2 diabetes, because although this association is clear in healthy adults, it hasn't been confirmed in people with type 2 diabetes, but we hypothesized that there would be the same positive association between the two. We also wanted to examine the effects of body fatness and aerobic fitness on age-related increases in people with type 2 diabetes. And we hypothesized that higher aerobic fitness would attenuate and higher body fatness would exacerbate the age-related increases in arterial stiffness. So this is a secondary analysis from baseline data from the larger randomized controlled trial, and that investigated the short and long-term efficacy of high-intensity interval training in people with type 2 diabetes. So the overarching inclusion criteria for the study were males and females aged 18 to 80 years old with a diagnosis of type 2 diabetes, including a HbA1c of greater than or equal to 6%. We excluded any people that were meeting the physical activity guidelines, had any conditions as per the American College of Sports Medicine's absolute contraindications to exercise, which is typically around severe cardiovascular disease or end-stage kidney disease. If people had any planned operations during the research period, or they had any physical conditions that limited their ability to exercise or was inappropriate for them to exercise. So individual assessments were conducted at the University of Queensland. Pulse wave velocity, carotid femoral pulse wave velocity, was used as the gold standard assessment of arterial stiffness. So before the participants came in, they were asked to avoid strenuous exercise, caffeine, alcohol, and tobacco, and well as maintain their normal medication regime in the 24 hours prior to testing. And then we also got them to complete an overnight fast of at least 12 hours before we did this test. So after 15 minutes of rest, we measured arterial stiffness using a sphygmocore XL, which you can see on the picture here. So basically, to measure arterial stiffness, this is a non-invasive way to do so. We put a cuff around the upper thigh, around the femoral artery, and then place a carotid tonometer sensor on the carotid artery. And then basically we just capture simultaneously the pulse waveforms that are occurring at those two sites, and the velocity of the pulse wave from the carotid artery down to the femoral artery is measured and calculated using the sphygmocore software. To measure aerobic fitness, participants completed a maximal cardiopulmonary exercise test. So after they did a bit of a warm up on the treadmill, people exercised in continuous 1 minute stages at an individualized speed and load until exhaustion, voluntary exhaustion. Pulmonary gas exchange was measured to assess peak oxygen uptake, both in absolute terms, but also in relative terms to body weight. And so from this test, we also got exercise capacity, which is based on their time to exhaustion, so how long the person was able to exercise for. To assess body fatness, we used a few different methods. The first was body mass index, just to get overall body adiposity. We also measured waist circumference using a tape measure to get information on central adiposity. And then we completed a DEXA scan, so a dual energy X-ray absorptiometry scan, which you can see on the slide there. And this allowed us to assess total body fat mass, which on this picture is in the yellow. So in terms of statistics, we ran two separate analyses. The first was Pearson's correlation coefficients to describe the associations between age, body fatness, and aerobic fitness with the arterial stiffness measure, that carotid femoral pulse wave velocity. We then ran multiple linear regression models to investigate the associations between all those variables, while also adjusting for sex and duration of diabetes, because these are known influences on arterial stiffness. So overall, we had 64 participants, 24 of which were female. The average age of our cohort was about 60 years old, with an average HbA1c of 8%, which means, I guess to put it into context for diabetes, the goal of HbA1c is below 7%. And on average, our cohort had diabetes for over 10 years, but the range was only a few months up to 30 years, so quite a large range. Our sample had elevated arterial stiffness with that carotid femoral pulse wave velocity measure, and that's compared to a population that is of the same age but has no cardiovascular risk factors. The mean waist circumference of our sample, which was 109 centimeters, is above the upper threshold for cardiovascular disease risk, according to the NHMRC. Our sample also had lower than average relative VO2 peak or fitness, which placed them in the 25th percentile for their age and sex, with a range of the 5th percentile all the way up to the 90th percentile. So our first analyses were the correlations. As you can see, the biggest correlation that we saw was in age, the strongest association. We also looked at the body fatness and fitness measures. Because waist circumference and relative VO2 peak were the indicators of fatness and fitness that had the strongest correlation with the pulse wave velocity, these variables were then selected to be included in the series of multiple regressions to represent fatness and fitness. So just to simplify the message from the regression models, overall for the initial model, which only included age, sex, and duration of diabetes, we found that for every 8.7 years increase in age, which was the standard deviation for our sample, there's a 0.6 meters per second increase in pulse wave velocity. When we added waist circumference to that model, that went up slightly to 0.66 increase in pulse wave velocity for every 8.7 years. When we added relative VO2 peak to that model, so not including waist circumference, it went down to 0.57 meters per second increase for every 8.7-year increase in age. And then when we included both waist circumference and relative VO2 peak, the increase in carotid femoral pulse wave velocity was the same when we included both. And that's because the waist circumference and the relative VO2 peak were going in opposite directions. And so they sort of cancelled each other out a bit. To look at that a little bit more clearly, we used the regression equations, to derive estimates of pulse wave velocity in 10-year age groups for different values of waist circumference and relative VO2 peak. So in this graph, hopefully you can see my mouse, but on the y-axis, we've got the carotid femoral pulse wave velocity, that arterial stiffness measure. And then we've got 10-year age groups along the x-axis. So in each color, we then have the waist circumference cutoffs, and they went up in 10 centimeter increments from 80 centimeters to 140 centimeters. So as you can see within each age group, just increasing your waist circumference by 20 centimeters results in a clinically meaningful difference in pulse wave velocity. So a clinically meaningful difference is 1 meter per second. And so that relationship held true throughout all of the age groups, where for every at least 20 centimeters, we saw a clinically meaningful increase in arterial stiffness. When looking at the same graph, but this time with the fitness measure, so the relative VO2 peak, we saw that with the higher levels of fitness, so up to 40 mils per kilo per minute, which is the relative fitness measure to body weight, if you only went down in fitness by 10 centimeters, so going from the green to this apricot color, that was again a clinically meaningful increase in pulse wave velocity. So as your fitness increases, we're seeing protective effects on arterial stiffness. And again, that rang true across all of the age groups. For this final one, we looked at both waist circumference and relative VO2 peak in the same equation, this time looking at just the average age of the cohort. So at about 60 years of age, how does waist circumference and fitness influence this relationship? So as you can see, we've got waist circumference on the x-axis here, and then within the colors, we've got the relative VO2 peak. So at a lower waist circumference, you can see that lower fitness is going to cause an increase in arterial stiffness. So as we increase fitness, we can see a protective effect on arterial stiffness at every waist circumference level. The same also goes in the opposite direction. So for lower fitness levels, for lower fitness levels, when we increase waist circumference, you can see that there is no longer that protective effect. So increasing in waist circumference is going to increase arterial stiffness as well. So overall, as a summary of the results, we found that age was a significant determinant of increased arterial stiffness, irrespective of sex and duration of diabetes. We found that higher aerobic fitness attenuated and higher body fatness exacerbated that age-related increase, but the effects were similar in magnitude, but in opposite directions. So when they were in a combined model, they sort of cancelled each other out. The mechanisms that underlie the association between fitness and arterial stiffness aren't fully understood, but it has been suggested that high aerobic fitness, which results from high levels of exercise, decreases calcium deposits in the aortic wall, which prevents unhealthy arterial remodeling and dysfunction. We also have seen that higher aerobic fitness is associated with improved metabolic risk factors for type 2 diabetes, including glucose metabolism and inflammation. And those have also been implicated in the pathogenesis of increased arterial stiffness. In terms of the mechanisms by which body fatness exerts a detrimental effect on arterial stiffness, there are three main pathways. So the first one is that excess adipose tissue leads to adipose tissue dysfunction and insulin resistance, which creates a bit of a lipotoxic state that reduces nitric oxide bioavailability and increases vascular tone, which results in arterial stiffening. Another mechanism may be the elevated levels of leptin that we see in both obesity and arterial stiffness. And that again impacts vascular tone, but it also increases sympathetic nervous system activation and elevates blood pressure, which therefore increases arterial stiffness. Finally, we've got osteocalcin, and that's a bone-derived hormone that modulates glucose metabolism and fat distribution, and it actually protects endothelial function and prevents atherogenesis. So low levels of osteocalcin have been seen in both obesity and are also associated with high arterial stiffness. So what does this all mean? Well, overall, lifestyle interventions which increase fitness and also reduce fatness are required to reduce arterial stiffness in people with type 2 diabetes. Where changes in fitness are the target, any modality of regular aerobic exercise should be encouraged, but some modalities like high-intensity interval training will provide more rapid improvements in fitness. Where changes in body fatness are the target, the current guidelines promote higher volumes of aerobic exercise for weight loss and weight maintenance. In saying that, the combination of diet and exercise is going to be most beneficial for changes in body composition and body fatness than just exercise alone. So including a diet intervention alongside exercise will be important. Something else important to note is that clinically meaningful increases in fitness are going to occur much more rapidly than changes in body fatness. And so increasing fitness may be a more useful early therapeutic target for reducing arterial stiffness in people with type 2 diabetes before the changes in fatness can be achieved. So in summary, our results provide preliminary evidence that age is a significant correlate of increased arterial stiffness in people with type 2 diabetes, with higher aerobic fitness attenuating and higher body fatness exacerbating that increase. A couple of limitations to note, the main one being that because this is a cross-sectional study, we're not able to determine causation, so future studies should be prospectively evaluating this association. Of course, this study is also a relatively small sample size, so the results should be interpreted with caution until we can replicate that in a larger sample. But overall, our study has provided this initial sort of indication that interventions which aim to improve cardiovascular health outcomes by reducing arterial stiffness in people with type 2 diabetes should target both fitness and fatness. Thank you all for listening and attending today. And of course, thank you to all of my wonderful collaborators from the University of Queensland, Griffith University and the Wesley Hospital in Brisbane. And thank you.
Hi, I'm Audrey Ajit from Victor Jackson Veterans Hospital, Sydney. Nice presentation. Congratulations.
Thank you.
First of all, probably we all know that diabetes accelerates arterial stiffening. My question will be, number one, have you looked at the males and females separately? Because we had a study last year that's been published, and apparently all these changes is more apparent in females than males. That's my first question. And number two, can you elaborate a bit more? Because I was a bit surprised looking at your correlation that you found significant correlation with the waist circumference, but not body mass index. Yeah, if you can elaborate, because as I said, our study population last year is like more general population, so she's not particularly looking at people with diabetes. But yeah, if you have any hypothesis behind it. Thank you.
So your first question was about males and females. So we didn't look specifically at sex differences in this relationship because we did have less females, so we didn't have enough of a sample to get both, which is why we included sex as a covariate in our regression models. So I don't actually have an answer to that question. Someone else may have done this study before, but I haven't seen that. So I don't actually know if there is a difference, but we just unfortunately didn't have the scope to do that for our sample. The second question was about the BMI and waist circumference. So yeah, I guess BMI, even though it's a really easy and accessible way to assess body fatness, it is limited in the fact that it doesn't differentiate between muscle and fat. So someone can be really muscular and have the same BMI as someone that has absolutely no muscle and a lot of fat, because muscle is actually heavier than fat. And so I guess it's probably somewhat not surprising that that didn't have as strong a relationship as waist circumference. And we've also seen in other studies that visceral adipose tissue, so the fat that is around the organs, is a significant cardiovascular risk factor. And so waist circumference is able to sort of delineate that, where body mass index isn't able to do that. But yeah, it is interesting that it was so much stronger and actually a significant difference, but that's probably the main reason why, I guess.
Thanks so much, Emily. We have Dr. Anastasia Mihalibu. He has a question. Thank you, Anastasia.
Thank you, Isabella. Emily, great presentation. Thank you. I'm very interested in one of your exclusion criteria was currently meeting physical activity guidelines.
Yeah.
Given that it's so low for Australia and the world, how many did you find you had to exclude, and how do we meet target? Because that's a key problem globally.
Yeah, oh, I wish I had the perfect answer for that question. Yeah, so I guess we probably actually didn't exclude a huge number of people. I don't actually know the full numbers. Our overall trial, we had 70 people, and we screened over 400 people for inclusion in the trial. Most of those people were excluded because they had contraindications to exercise. But there were certainly probably about maybe 20% of those people were meeting the physical activity guidelines. Inactivity in Australia in people with type 2 diabetes is at 55%, which is above the general population of inactivity. So I guess that's sort of a question that we're trying to answer now, and where my research is sort of leading is, how do we get people actually exercising? We're at the point now in exercise research where we're blue in the face saying that exercise is beneficial, exercise is medicine, but people have to do that. People have to take that medicine to be able to get the benefits. And I think where my PhD sort of initially led me was I did a randomized control trial in high intensity interval training. And I used a low volume, which is about a third of the time of the current guidelines. And people were still able to get about the same benefits that they did with the current guidelines, which is 210 minutes of exercise per week for diabetes. But they were able to get that in only 78 minutes per week. So I guess that's one barrier that we're getting over, is we always hear people don't have enough time to exercise. Everyone's really busy, and exercise usually goes to the bottom of the list. But that's sort of one way that we're trying to approach, is that using this sort of time efficient method of exercise to get people to just squish it into their week where they can and participate as much as they can without giving up some of the benefits of exercise.
Thank you.
Thanks so much, Emily. In the interest of time, we might just have time for one more question. And if no one else has a question, I'd like to ask the last question, please, Emily. So I'm really sorry if I missed this, but did you measure blood pressure in your participants? And did you see any effect of blood pressure at all in the pulse wave velocity values that you measured? Because in your regression model, I didn't see that you corrected for blood pressure. Is that right?
No, that's right. So we did measure both brachial and central blood pressures, and they were initially included as covariates in the regression model. But as we were trying to simplify that and take out variables where we could, blood pressure didn't influence any of these relationships. So we didn't include it as a covariate, but yeah, that as well as antihypertensive use as well. We use that as a covariate, but again, it didn't impact any of the relationships. And so we took it out just for simplification of the models.
Perfect. Well, thank you again, Emily, for your time and for your brilliant talk. Thank you so much.
Thank you.
But I'd like to introduce to you our second speaker for today, Dr. Christopher Stanley. For over a decade, Dr. Chris Stanley's research has focused on the function, structure, and signalling of resistance arteries. Chris received his Bachelor of Science degree with honours in pharmacology and physiology from Leeds Metropolitan University in the UK, and his PhD in the same discipline from the University of Nottingham. Chris then undertook postdoctoral studies with Professors Kim Dora and Chris Garland at the University of Oxford, where he investigated the effects of human disease on microvascular structure and function. In 2014, Chris moved to Australia to work with Professor Roland Stocker at the Victor Chang Cardiac Research Institute on a project investigating tryptophan-induced arterial hypertension. Oh, sorry, I got that wrong. It's tryptophan-induced arterial relaxation. That's really important. This work led to the discovery of a novel tryptophan-mediated mechanism that governs arterial tone and blood pressure. For this work, Chris was awarded a Society for Free Radical Research Young Investigator Award, a Sydney Cardiovascular Symposium Rising Star Award, and the University of New South Wales St Vincent's Clinical School Publication of the Year Award. Chris now leads the microvascular research unit at the Heart Research Institute in Sydney, where he continues to work in this area. So it's with great pleasure that we welcome Chris today, telling us about tryptophan metabolites and the control of arterial tone and blood pressure. Thank you so much, Chris.
Thank you for the introduction, Isabella, and thank you to the CVRN for allowing me to speak today. So as Isabella said in the introduction, I'd like to detail to you today the discoveries that we made regarding tryptophan metabolites in the control of arterial tone and blood pressure. And this really centers around inflammation and specifically in septic shock. And despite the fact that I'm going to detail how we want to inhibit this pathway to you today, hopefully next year I can come back and talk to you about how we want to stimulate this pathway in the treatment of hypertension, as I've got some pilot data on the go that looks quite promising in that vein. So without further ado, then I'll move into septic shock and the discovery of this pathway. So septic shock, as you are undoubtedly aware, is an unmet clinical need. It's caused by a microbial invasion and then a systemic dysregulation of the innate immune response. In Australia and New Zealand in ICUs in 2012, it was reported that 12,500 patients presented with severe sepsis, and this had a mortality rate of 1 in 5 for those presenting with septic shock. The Australian Sepsis Network reports 15,700 sepsis cases and 3,000 lives lost per year. So this is a very real national problem, and it's also a very real global and international problem. The loss of life generally occurs on account of an impaired organ function and hypertension. And this hypertension really occurs through uncontrolled vasodilation of the peripheral arteries. So if we're going to treat septic shock and we're going to treat this hypertension, then we really need to know more about vasodilation of peripheral arteries. So this generally occurs in the endothelial cells, and the vascular endothelial cells are extremely good at producing compounds that dilate arteries. So here you can see that the endothelium is able to produce prostaglandins from arachidonic acid metabolism. It's also able to produce nitric oxide and hydrogen peroxide. These molecules are able to move from the endothelium to the smooth muscle, where they interact with their target proteins, such as protein kinases, and ultimately they hyperpolarize vascular smooth muscle cells by opening ion channels. Most notably, or most well researched, are potassium channels, and they then relax the arteries. There's also another mechanism that I worked on during my time in Oxford with Kim Dora and Chris Garland, and this is electrical conductivity through opening of potassium channels in the endothelium, and then the changes in membrane potential spread from the endothelium into the vascular smooth muscle. And again, this is a very powerful mechanism of arterial dilation. Despite this research into mechanisms of arterial dilation, when researchers have taken this knowledge to clinical trials, so they've investigated these pathways such as the nitric oxide pathway or the prostaglandin pathway through COX inhibition or the potassium channel inhibition, in the treatment of septic shock, these clinical trials have often failed or showed non-favorable or transient improvements. So what we're left with is the clinical treatment of septic shock still relies on overcoming the hypertension via addition of vasopressin agents instead of preventing the hypertension from occurring. Now, you might ask, what's wrong with overcoming hypertension through addition of vasopressors? Well, this itself can lead to microvascular dysfunction. And we're not actually resetting the vasculature and allowing it to do its own thing, which it's incredibly good at. What we're doing in this instance is we're overriding the system, which itself causes problems. So as Isabella said, when I came to Sydney, I was incredibly fortunate to work with Professor Stocker, who at this time was proposing that maybe we didn't know everything that we needed to know about arterial dilation, especially in sepsis. And he'd conducted work in cerebral malaria, which also has a hypertensive phenotype. And he wondered whether there was a role for an as yet unidentified molecule or pathway that potentially mediated this hypertension and vasodilation in sepsis. And he'd drawn from examples in nitric oxide and its ability, its functions within the body. So we know that nitric oxide can be formed through nitric oxide synthases. There's several varieties of nitric oxide synthase, some which are induced on inflammation. So we were looking for something that was potentially induced via inflammation, and that potentially the molecule that was induced through inflammation had immune regulation, antimicrobial, neurological roles, and maybe functioned as an antioxidant. And through his work in cerebral malaria, Professor Stocker stumbled upon IDO1, or he was interested in indolamine 2,3 deoxygenase 1, which is the enzyme which metabolizes tryptophan. And through his work in malaria, he realized that it had immune regulation roles, it had antimicrobial roles, and interestingly interacted with nitric oxide and nitric oxide synthase. But what we didn't know at the time was, did it cause arterial relaxation? So looking into the literature, when we wanted to see whether there was any precedence for the role of IDO1 in sepsis, we found a paper that was published in 2009 in the Journal of Immunology, which looked at IDO1 knockout mice and their response to an aggressive LPS model of sepsis. And what we saw was that there was an increased survival in these IDO1 knockout mice. Similarly, if you pharmacologically inhibited IDO1, again, you got an increased survival. Now, the researchers of this paper hadn't investigated blood pressure or arterial function. Instead, they'd investigated the immune response, and they'd seen several favorable immune outcomes. We were interested in blood pressure. So we wanted then to use a model. Sorry, I'm struggling to get my video to work. Sorry, my video's not working, so we'll skip that. So we wanted to use a model to see whether IDO had a role in causing vasodilation and arterial relaxation in a model of sepsis. So we used our LPS model of LPS mouse model, and we injected mice with LPS, and then we showed that we could relax arteries using L-Tryptophan. So this was the first time that we'd started to show that L-Tryptophan caused arterial relaxation in mouse mesenteric arteries, in these little resistance arteries that control blood pressure. So we wanted to know then more about tryptophan metabolism. And we know that tryptophan is metabolized by IDO1 and it forms informal kynurenine. For IDO1 to be metabolically active, it requires reductive of the heme within the enzyme to do this. So generally the heme is reduced from the Fe3 to the Fe2 conformation, and then this allows metabolism of tryptophan down the informal kynurenine and kynurenine pathway. Once you start this, then you get a range of metabolites such as kynurenic acid, L-kynurenine, and a whole bunch of other kynurenines. So we wondered then if IDO was involved in this arterial relaxation response, was tryptophan being converted to one of these compounds which was causing the relaxation? So to probe this, we went back to the mesenteric artery and we constricted our artery using norepinephrine. So this is now wire myography. You previously saw pressure myography. And what we see here then is that addition of L-tryptophan relaxes or reduces the tone on the artery. And if we take an IDO1 knockout mouse, we can inhibit that response. So this now shows that tryptophan through metabolism, IDO1 mediated metabolism causes arterial relaxation. We can graphically show this, and we can show that it happens in whichever artery we choose to take from the mouse. So it happens in the resistance arteries, and it also happens in the larger arteries. We then looked at the renal arteries, and we see here that IDO1 is expressed in the endothelium of LPS treated mice, but it's not expressed in the endothelium when we do the isotype control. We also show that in an IDO1 containing mouse that we get a drop in blood pressure upon LPS exposure, and if we gene delete IDO1, we can inhibit that drop in blood pressure. And similarly, if we pharmacologically inhibit IDO1, we can again preserve blood pressure to some extent. So this really suggested that IDO1 was important in converting tryptophan to a metabolite which caused arterial relaxation. So first in that sort of cascade was this informal kynurenine, which I mentioned earlier. So we tested that for its ability to relax arteries. And we saw that in LPS treated or in naive mice, informal kynurenine was not able to relax arteries. So this suggested that the arterial relaxant was being produced somewhere here, potentially by an unknown metabolite of tryptophan breakdown. As I said to you before, IDO1 needs reducing before it can become catalytically active. So we reduced it using superoxide or its biological reductant cytochrome B5. And you can see here that we only get a peak in using isolated IDO1 enzyme around this informal kynurenine. And we characterize that to be informal kynurenine. And as I previously showed, we know that this isn't vasoactive. Some work had come out of Shane Thomas's lab at the time, which intrigued us, and it showed that you could oxidize IDO1 using hydrogen peroxide. And this led to the formation of this compound OIA. So we tested OIA and we showed that OIA wasn't vasoactive, but intriguingly, we got this huge peak here. When we isolated this peak and we added it to our vascular preparation, we showed that this peak was able to cause beautiful dose-dependent arterial relaxation. We then characterized this compound to be cis-tryptophan-derived hydroperoxide. So cis is based on its isomerization, the W stands for tryptophan, and the hydrogen peroxide is this OOH here. So this was great. We've done all this in enzymes and isolated artery, but now what we wanted to show was that cis-WOOH, or this tryptophan-derived hydroperoxide, existed in arteries. So we went to the abattoir, we collected a bunch of pig hearts, we isolated the carotid artery, we isolated the LAD artery, and we incubated with interferon gamma. We then added 15 labeled tryptophan, 15 N2 labeled tryptophan, and we were looking for 15 N2 labeled cis-WOOH. And what we found was that we could dose dependently increase cis-WOOH production in these arteries. We also knew that hydrogen peroxide was important for this, for the formation of this chemical. So we needed to show, or we wanted to show as well, that hydrogen peroxide again went up with interferon gamma. So now what we've shown is that we can produce this novel compound and that it's vasoactive, but we don't know how it's vasoactive. So we went back to nitric oxide and we wanted to look at how nitric oxide worked to see whether cis-WOOH worked by a similar mechanism. I'm not showing data for it today, but we tested cis-WOOH and its ability to stimulate the receptor for nitric oxide, and that being SGC. And that showed that cis-WOOH did not interact with SGC, or it did not stimulate SGC. Some work had come out of Phil Eaton's lab in the UK at the time, showing that hydroperoxide could maybe bypass SGC and directly activate protein kinase G. And this was through a cysteine disulfide bond formation. A disulfide bond formation at a cysteine residue within PKG. So we sent our compound to Phil and we asked for him to test to see whether cis-WOOH activated PKG in this manner. And activation is typically shown as a dimer of the PKG protein. And we showed that under a range of different conditions, cis-WOOH was able to dimerize PKG, and importantly, when the cysteine residue that's involved was mutated to a redox inactive serine, we got no dimerization of PKG, suggesting that cis-WOOH was oxidatively activating the protein. Again, this was isolated enzymes, so we looked then to look at, did this happen in an artery? Could we do this with tryptophan, and could we dimerize PKG? And you can see here that in isolated mesenteric arteries, we do indeed get a dimer formation. To take this back to the whole animal then, luckily Phil had developed a cysteine to serine mutant mouse, which was redox inactive. So this mouse has a high blood pressure phenotype. It is protected against sepsis to some extent. And you can see here that when we looked at the effects of tryptophan being infused into this mouse, in the wild type, we've got a drop in blood pressure that sort of recovered, but then maintained a bit of a decrease over the next few minutes. However, in the PKG mutant, there was no real significant alteration in blood pressure. So what we've shown now is that cis-WOOH is able to oxidize PKG, but we still don't know how it's hyperpolarizing the vascular smooth muscle. So work that I'm currently conducting at the moment is fleshing this out. So just some unpublished data to share with you, where I've looked at, upon IDO expression, we do indeed get a hyperpolarization of the vascular smooth muscle, which suggests that we do have opening of ion channels, presumably potassium channels, and that if we constrict arteries and at the same time we measure the membrane potential of the smooth muscle, we can hyperpolarize the vascular smooth muscle with low concentrations of tryptophan, and that this is typically mediated through the BKCA channel. So going back to our arterial relaxant assay, we add tryptophan to the vessels, and we show that in the presence of a BKCA channel inhibitor, we can prevent arterial relaxation. So in summary, we've shown that tryptophan can be converted to a novel hydroperoxide derived from tryptophan, and this requires hydrogen peroxide and singlet oxygen, which I've not touched on today, but it's published in our paper. I'd ask anybody to go and read it. It's incredibly interesting. And the cis-WOOH then oxidizes PKG, and that activates the BKCA channel, leading to arterial relaxation and hyperpolarization. In an effort to inhibit this pathway to potentially offer therapeutic scope in sepsis, we looked at enzymes which are almost professional in their ability to detoxify hydroperoxides. And a group of these enzymes are called peroxiredoxins. So peroxiredoxins become dimers as they neutralize hydroperoxides, and then the peroxiredoxins themselves are reduced back to monomers so that they can participate again in this redox cycle. So what we did, we used a ranafen, which inhibits this process, so then you get an accumulation in the peroxiredoxin, and the peroxiredoxin becomes inactive. And what we showed under physiological conditions was that if you do this with a ranafen, you can stimulate arterial relaxation, suggesting that under physiological conditions, peroxiredoxins play a role in maintaining arterial tone, and that this arterial relaxation through inhibiting this pathway is dependent on oxidation of PKG. However, when we came to do this assay in our LPS treated mouse, we showed that we didn't get any arterial relaxation through inhibiting this cycle. And that was quite surprising to us, because we expected the peroxiredoxins to have some control over our cis-WOOH. So we went to an enzyme assay again, and we looked for the ability for cis-WOOH to cause peroxiredoxin dimer, indicative of cis-WOOH being, or cis-WOOH interacting with the peroxiredoxins. But we didn't see dimer at appreciable concentrations of cis-WOOH. Or if we did see dimer, they weren't forming that well. Not when we compared to hydrogen peroxide, which is potentially what we assume was being scavenged by peroxiredoxins under the physiological concentration.
Sorry to interrupt, Chris. We are running a little over time, so if you could please wrap up in the next minute, please. Thank you so much. Sorry about that.
No worries, thank you. So in some experiments performed by Kat Walhuter, I believe is online, we then looked at the in vivo relevance of this. And what we did was we infused mice with tryptophan to look for peroxiredoxin dimer. So what we saw was we looked at our target protein first, and we showed that infusion of tryptophan, which we believe occurs in sepsis, causes PKG dimer. We observed that, but we saw that there was no appreciable difference in peroxiredoxins. So it suggests that our cis-WOOH is being formed, but it's not interacting with peroxiredoxins, which is typical of hydroperoxide. So we don't know yet what scavengers or metabolizes cis-WOOH, if anything. And I've kind of just covered that in my summary. So that just leaves me then to thank the members of my lab that have contributed to the most recent mechanistic work, the members of Roland's lab that contributed to the bulk of the data that I've shown you, and similar to staff at the Victor Chang and our collaborators globally. Thank you. Sorry for running over time. I was completely oblivious to the time.
Thank you so much, Chris. That was a wonderful talk. Thank you. And thank you so much for sharing those really intriguing and interesting results. It's very exciting work. I have, someone has a question already in the chat box, and I might just read that out. So Dr. Purgeld asked, Chris, does this have any implications for non-sepsis related cardiovascular disease treatment?
So yes, so we found that this pathway is also present in atherosclerosis. So we get IDO expression in the endothelium in models of atherosclerosis. And I've also got some pilot data suggesting that this pathway is present in hypertension. So going forward, I'd like to look at how we can potentially utilize this pathway in atherosclerosis or hypertension as a potential treatment, or to understand why it's not functioning properly and hypertensions may be occurring.
With this tryptophan-mediated relaxation, how does that compare with, like, say, the nitric oxide relaxation? Because we know the half-life of nitric oxide is so short in our bodies, and therefore all the medications that targeting nitric oxides haven't been all that successful in the antihypertensive department. So what do you think about this mechanism? What's that time sort of duration like, and whether that's going to be useful?
Well, I know from the vessel function studies that once you relax an artery through this pathway, it doesn't tend to recover. So it's not that it doesn't recover, but it has a more sustained response. So if you add something like acetylcholine, you often get a relaxation, and then the artery starts to recover from that relaxation and return to a constricted state. When you activate this pathway, the relaxation actually sticks around quite a bit. In terms of the chemical composition of cis-WOOH, it is stable in biological buffers to a reasonable degree. It's certainly a little bit more stable than nitric oxide. But as to its stability in a biological matrix, we haven't determined that yet. So we don't know how quickly an artery would metabolize it. So the relaxation might stick around a little bit, because once you oxidize PKG, it takes a little bit of time to reverse that. But then again, the compound itself might stick around. We just don't know how it's metabolized as yet.
Great, thank you so much for that. All right, so thank you once again to Dr. Chris Stanley and also to Dr. Emily Cox for speaking with us today.